May I start now? Yep. Thank you so much for the organizing committee. I'm Suda from the National University of Malaysia. As the topic suggests, it's called AMRColab. So basically, we got inspired by Collabville, and then we do a Google Collab on AMR. So we're focusing on AMR genome assembly. So probably you guys, I'm not too sure whether you guys know or not. Antimicrobial resistance is something is very serious. And if you look at Lancet report, it's saying that more than 39 million will be died by 2025 if no action is taken. World Bank data as well, also saying that it will cost a lot of money and drove a lot of people to poverty if you are not doing good. The good thing, the whole community around the world, they are started doing surveillance and doing some actions on it. And what happens usually is you have your dynamic samples, and then after the microbiologists will culture the pathogens and then check whether they are actually pathogenic or not. And then if they are actually like AMR, we'll actually proceed with whole genome sequencing. The reason why they want to do whole genome sequencing, they want to know what are the new mutations, are the new genes that actually happens in the new strain that you have. Then it passed to bioinformatics people. So we help with the sample extraction and then run the sequencing. And then you perform the bioinformatic analysis and then you get the result. For us, this is normal. However, I'm dealing a lot with people that don't know how to code. So a lot of public health practitioners, they don't know how to do coding. So when they see this kind of thing, it's hard. But for us, this is simple. Yeah. But that's the problem I'm having because I'm back in Malaysia. So it's a low middle income country. A lot of students of us don't know how to code. Another problem is we don't have a good machine to run. So without a machine, and some of them is just Microsoft Windows, so running the codes will take them hard to do. So that's another problem. So lucky enough, we have AlphaFold, and thanks to Sergey and Martin Senneiger came out with Collabful. Then I'm thinking, is it possible for us to develop a Google Colab for AMR genome assembly? The idea is simple, but it was very useful for our community in low middle income country. So the idea is you have your PC, and then after that you just connect to Google Colab. For those of you that don't know what Google Colab do, it's you have access to Google Colab machine for about 12 hours, and that machines usually is okay. Sometimes you get GPU, sometimes you get TPU, but that computer is for you for a while. And then it's free. You don't need to set up everything, and it's totally free. So what happened here is a non-bioinformatic people can start to upload the data, their genome assembly, and then after that they can perform the bioinformatics too. And they just need to provide some simple thing like file name, what type of sequence they have, and what organism they have. And then after that, they can download the result they have. So this is just a demo. So probably everyone here is familiar with Google Colab. So it's click. So it's easy for them to do the work. Yeah. So we have like 5 modules at the moment. So we have the genome assembly module. This one, it takes a while to run because normal computers still take a while to do genome assembly, but we were asked by our reviewer to do it, so we add it. And apart from the genome assembly, you also do AMR gene prediction. And there's also, we have added AMR Finder Plus and REST Finder that's commonly used. We also work with the Phage team where we've joined their team where they have a software called harmonization where you can harmonize the AMR detection result in the report. And then after that, you need a visualization module where it tells them how to visualize the result. You can use Pathogen Watch and MicroErect. One thing to say, all of this module are you can access this individually, so you don't need to follow every single thing to use it. You can use any one that you want. The end output will be like this. So we've done workshop. Yes, the first workshop was 2022. 2023, we did another one. Most of our participants, they don't have a lot of bioinformatic knowledge. Yet we got a lot of good feedback from them. They think it's very good, not even for non-bioinformaticians. Very informative, very easy to get. A lot of them actually like our output, confidence using it, and started doing some bioinformatics too. In workshop, we also they also started to saying they're using the method now. We've published this last year in Microbial Genetics, and we have a GitHub page that basically just provide the link to all of the Google Colab that we have produced. It's licensed under this one, Creative Commons. And then we have a manual. And I need to thank Fage for helping us, my team in Malaysia. And this also involves someone from Argentina and also Vancouver, Professor Christine Origo, my PhD boss at that time. Workshop participants, manuscript reviewer, and my grant at the moment. Thank you so much.